All right, it's time for our fourth and final segment on erythropoiesis. Currently, we are discussing some problems with red blood cells. In particular, I'm focusing on this last segment on different types of anemia. And if you have your slide that I told you to print up that has the different color coding, you'll notice that I'm still on blood loss, but this time I'm going to discuss hemolysis, acquired hemolysis. Now, acquired hemolysis can be from basically you being bitten by a rattlesnake whose venom has hemolytic properties, or we can have hemolysis through acquired means like incompatible blood transfusion. We also can consider hemolytic disease of the newborn as a type of hemolysis, and some drugs can also cause hemolysis. So in this case, the red blood cells that are made are normal. Everything about them is normal. It's just that they are being attacked and lysed very, very rapidly. Moving on to a type of blood loss would be through having thalassemia. Now, in this case, this is still hemolysis, so it's still blood loss, it's hemolytic, but it's not acquired. In this case, it's inherited. Now, there are two broad classifications of thalassemia. It's either alpha thalassemia or beta thalassemia, and they are named, and listen up, they're named for the gene that has the mutation. So, for example, if you have alpha thalassemia, then that means you have a mutation in one or more of your alpha genes that code for the alpha globin chains. And those alpha genes are found on chromosome 16. If you have beta thalassemia, it means you have a genetic mutation in one of your beta globin genes, which are found on chromosome 11. Now, the thalassemias can be severe to very mild. And what happens is, let's pick beta thalassemia for an example. Look at the red squares that represent the genes. Let's say you have a mutation in both copies of your beta globin genes. Then that means you would fail to make the beta globin chain. It would not be there. So those little red circles, put an X through them. That means all you would have for assembly would be the alpha globin chains, right? So it's the unaffected chain that accumulates and starts to bind with itself. So you would be making improper hemoglobin with just alpha globin chains. Now, it turns out that this inappropriate complex of alpha globin chains precipitates out of solution and causes hemolysis of the red blood cell. In the case of alpha thalassemia, it would be the beta globin chains that accumulate and become disastrous. Now, look at the number of alpha globin chains. There are 4 copies. Let's say a person has a mutation in just one copy. Do you think they would present with any issues? No, because there's so much redundancy. Students often ask, well, why are there so many copies of the alpha globin genes? And I'll remind you, the alpha globin chain is required not just for embryonic, but fetal and adult hemoglobin. So it's a very important globin chain, and it's a very important gene. So having redundancy is a protective effect. What if a embryo has a mutation in 2 genes? Again, they might not present with any significant severe problems because they still have 2 normal genes. When we would start to see a problem with the alpha thalassemia is if 3 or more of the genes are mutated. And if all 4 are mutated, then this is going to lead to death in utero. This is not compatible with life. Now, despite the type of thalassemia, we're going to see the red blood cells are hypochromic and microcytic. they're going to have the same kind of profile as iron deficiency. So MCH goes down, MCV goes down, MCHC also down. Obviously, with hemoglobin molecules that are not made correctly, this can lead to organ failure because of can't carry oxygen, obviously, very well. And the red blood cells that are being destroyed or hemolyzed, this means there's going to be an increased workload for the spleen and liver, and those organs become enlarged. So we see splenomegaly and hepatomegaly. Now, the reason why the unaffected chain is a problem, again, it only has itself to form a quaternary structure with. So it accumulates inside the red blood cell, and this is going to cause Heinz bodies, as they're called, where that unaffected chain kind of precipitates out of solution. Now, in the case of alpha thalassemia, when you're not making any of the alpha chains, and in the embryo, of course, this is when it would first present, and the embryo would only be making the epsilon chains, and they would be accumulating with each other, and this is called Bart's hemoglobin. It's not compatible with life. Again, in utero, fetal, or embryonic death. Now let's talk about thalassemia, the beta thalassemia in particular. Again, if both genes are mutated, this is called thalassemia major or Cooley's anemia. And again, it's a shortened red blood cell lifespan. The alpha chains accumulate. This leads to profound hemolysis. With profound hemolysis, of course, we can complete our reflex arc. More erythropoietin would be released. This would stimulate the hemocytoblasts and stem cells to push the daughter cells into more red blood cell synthesis. And it's all for naught because it's a genetic mutation. This is going to lead to an expansion of the medullary spaces in the long bones, and to the point where there's very little cortical bone that's left, and this means that the person can have skeletal abnormalities, quite painful, prone to skeletal fractures. And with the profound hemolysis, of course, there's release of iron, then that means there's going to be greater iron storage and iron overload in the hemosiderin, and we know that that's toxic. And sometimes with this type of anemia, the Cooley's anemia, we can actually see the presence of fetal hemoglobin. And although that has a higher affinity than adult hemoglobin, it actually is still kind of a neat thing to see persist in the patient, because again, that would be the alpha 2 and gamma 2 type of hemoglobin. Higher affinity, less likely to give up oxygen, but at least it's better than nothing in the patient. So, thalassemia is mostly associated with people of Mediterranean descent. That's why I'm showing this here. And I am going to move on to more types of anemia. The next one I want to highlight is hereditary spherocytosis. Again, it is still blood loss. It's a problem with hemolysis. In this case, it's inherited, intrinsic, not acquired. And in this case, it is a problem with the red blood cell membrane and maintaining it. The cells are initially made correctly. They are normochromic, normocytic, but over time, the red blood cell membrane is lost. It kind of blebs away. So the MCV gradually decreases. Now this means that the hemoglobin is now going to be in a smaller and smaller volume, which means MCHC increases. So I want you to think of the end portion of this disease. For testing purposes, hereditary spherocytosis leads to normal MCH, decreased MCV, and increased MCHC. It is the most common inherited blood cell membrane disorder. And we can treat this with splenectomy. remove one of the organs responsible for removing red blood cells. Here's why. Here's why it works. Again, the liver and spleen have reticular fibers. And if the red blood cells cannot move through those reticular fibers, they get hung up and the macrophages ingest them, phagocytize them. So with hereditary spherocytosis, the cells have poikilocytosis. They're spherical, and they can't shimmy their way through the reticular fibers. So they get removed. That's the hemolysis part. Well, if you remove one of the organs that have those reticular fibers, then you aren't going to have as much red blood cell hemolysis. So we can remove the spleen safely, and this is effective. We don't remove the liver, right? The liver does 500 functions for the body. You can live without your spleen, but you can't live without your liver. All right, another type of hemolysis disorder, again inherited, is sickle cell anemia. Now this is again a genetic defect, just like hereditary spherocytosis and thalassemia, but in this case, in the amino acid position number 6, there has been a mutation that leads to a substitution. Instead of glutamate in position 6, a valine is inserted, and this is shown in the top right sequence. Now, glutamic acid is a polar amino acid. Valine is nonpolar. And you remember me telling you that the amino acids with their functional groups dictate the chemistry of the protein. So we are, with such a little change comes dramatic effects. What happens is, with sickle cell disease, when the red blood cells come into conditions that have low oxygen content, then the beta globin chains start to link together and they form these stiff rods inside the red blood cell. Again, the mutation occurs in the beta globin gene. Under low oxygen conditions with sickle cell disease, the beta globin chains form these rod-like structures, causing the red blood cells to sickle, as shown in the animation in the middle here. And these sickled red blood cells can block and occlude blood vessels, which is shown down here at the bottom picture. So the sickled cells, when someone has sickle cell disease, the type of hemoglobin they have we call HbS. Instead of saying HbA, which stands for adult hemoglobin, we say HbS. And this means that we now recognize that one of the beta chains has valine. Now, the red blood cells are normochromic, normocytic, but they will become poikilocytotic when there is an episode. So again, under normal conditions, the red blood cells have normal MCH, normal MCV, normal MCHC. It's when they have an episode that the cells change their shape and become sickled. Now, when these rod-like structures are made, it can lead to hemolysis because the red blood cell membrane can be easily ruptured by these rod-like structures. Let's continue this discussion. Okay, so here are pictures of chromosome 11. Again, we have 2 copies of the beta globin genes. We express both of these genes in a codominant fashion, meaning we use both. It's not just one or the other that like we usually associate with true Mendelian genetics. So we use both. So in a person who makes adult hemoglobin, of course we would recognize that the beta globin peptides or proteins would be coming from both the beta globin genes. So our genotype, as shown in the first dash, is if our genotype is HBB and HBB, then our mature erythrocytes would be filled with properly made adult hemoglobin. But if our genotype is HBB and HBS, then we are going to get a mixture of hemoglobin. We still have one functional gene and one mutated gene. So we would have a red blood cell that would have both normal adult hemoglobin as well as the sickled cell or sickled type of hemoglobin. It'd be a mixture. And clearly, that person would have fewer problems, right? If, however, the person has a mutation in both genes, then the genotype would be HBS, HBS, and the mature red blood cells would only be making the sickled form of hemoglobin. So it would be the normal alpha globin chains along with the sickle beta chain. Now, you can go to this website and actually learn more about this genetic mutation if you'd like. We can easily detect the change in the protein by using what's called gel electrophoresis, and you will be taught how exactly this works at a future date. But until we get there, it's really easy. We draw the patient's blood, we load the blood sample in this, it looks like a jello slab, and then we apply an electrical current. And if you look at the bottom right picture, you can see that a person's normal hemoglobin versus a person with sickle cell anemia versus sickle cell trait. The trait means they have one bad copy of the beta globin gene and then the mutated copy. And you can see that the sickled hemoglobin versus adult hemoglobin would migrate through the gel differently. And this is the way that we can diagnose if a person has sickle cell trait, which means one bad copy, versus sickle cell anemia or disease, which means both the beta globin chains are mutated. So again, the heterozygous individuals have sickle cell trait. They would have a milder form of the disease, and they would only have an episode if they had hypoxia. It's usually not life-threatening. And interestingly, in African Americans, there is a high population rate of this trait, having one bad copy. And for geneticists, whenever we see a population of people having a high frequency of what would be considered a deleterious, bad mutation, it makes us stop and scratch our heads and say, there must be somehow a protective effect or an advantage if a person has just one bad copy of this gene. And it turns out that people with sickle cell trait who live in regions affected by malaria, they have resistance to it. And I'm going to talk about that on the next few slides. People who have both copies mutated are homozygous individuals and can only make the sickled form of hemoglobin. So they would have their normal alpha globin chains, but only the sickled beta globin chains. They would have crises, they would have episodes anytime that their tissues have low oxygen content. Again, it's the low oxygen content that causes the beta globin chains to start to assemble in these rod-like structures. So literally just falling asleep, we aren't inhaling as much oxygen, we aren't breathing as much. That alone can cause someone to have a crisis. Anytime they get sick, if their respiratory tracts are compromised, it could lead to a crisis. This is life-threatening, and it's very painful, and it can be a positive feedback effect. If there's a crisis, the red blood cells sickle, they block the blood vessels, the tissue becomes increasingly hypoxic, it leads to less oxygen and more sickling. Now, we can give people a drug, hydroxyurea, and this helps convert their hemoglobin synthesis back to the fetal form. And again, that is great because then fetal hemoglobin is alpha 2, gamma 2. And although this has a higher oxygen affinity, it still is better than having a crisis. Now, let's talk about malaria. Malaria, of course, you're seeing the vector for malaria. It is one of the phases for this plasmodium is that it goes from human into an insect vector. So the parasite multiplies and undergoes many different stages in its life cycle. It needs to move to the patient's liver to complete some of this, and then to the human red blood cell. And what happens is it causes the cells to rupture, and this can lead to reinfecting of new red blood cells, and this leads to high fevers. So malaria actually means the word bad air, and it was thought that people who got malaria, it was something in the air. It wasn't the air itself, but what was flying through the air was the vector for the transmission, the female Anopheles mosquito. So the female, of course, bites someone who's infected, gets some of the blood, and then goes and bites another person. And when a mosquito bites you, they release some of their saliva to numb you so you don't feel them piercing you. And when the female mosquito does this, releases her saliva, some of the blood from the previous victim now infects the new person. So now this parasite infects the new person's red blood cells. And it's a perfect way for a plasmodium, a parasite, to evade our immune system by hiding inside our red blood cells. So it goes virtually undetected unless you have sickle cell trait. And then in this case, having sickle cell trait can actually give you an advantage to fighting off this infection and not succumbing to it. Now, what do I mean by that? Well, when the parasite hides in our erythrocytes, or in a person specifically who has sickle cell trait, well, the parasite starts using some of the oxygen in that red blood cell. And again, a person with sickle cell trait is going to be making some of their hemoglobin with the sickled form of the beta globin chain. As the parasite starts using some of the oxygen in the red blood cell starts to have hypoxic conditions, and it will start to sickle. And if it sickles, it will have less ability to get through the reticular fibers in the liver and spleen. And so it means then the macrophages are going to remove those sickled red blood cells that are infected with the with the plasmodium. And this means that the very red blood cell that the plasmodium is trying to hide in gets eliminated. It means you're getting rid of the parasite. So it's easier for the person to fight off the infection. And therefore, that's the reason why we see so many people of African American descent carrying one copy of the sickle cell mutation. So, because they lived to fight another day, because they fought off the malarial infection, they were then able to reproduce, right? And so the mutation gets passed on to the next generation. So again, the advantage in fighting malaria is having sickle cell trait, but there is no advantage at all in having sickle cell disease. And if you want to watch a PBS special or a YouTube video on the Anopheles and how it is a vector transmission and how having sickle cell trait is protective, I encourage you to watch these videos, especially the PBS one. It's only like 11 minutes long, which is drastically shorter than all of these lectures, right? Now, the last type of anemia that I want to talk about is still, again, hemolytic, is G6PD deficiency. Now, this is still genetic. It is going to lead to an inherited hemolytic blood loss. It is the most commonly inherited enzyme defect in red blood cells, as opposed to red blood cell membrane defect, which was hereditary spherocytosis. This is X-linked, and so therefore you would predict men would have more of a problem than women, because they only have one X chromosome. There are many different types of mutations that can occur in the gene. So there are many genetic variants, and my video won't play because of the software that I'm using, but I'm showing you a picture of Hannibal Lecter where he said, a census taker once tried to test me, so I ate his liver with a nice with some fava beans and a nice Chianti. You remember that part from Silence of the Lambs? So super freaky. Well, why do I have Hannibal Lecter on a slide talking about G6PD deficiency? Well, here's the reason why. First, what exactly is the problem with this genetic mutation? What happens is this enzyme that is mutated is an enzyme that helps convert the methemoglobin, remember the +3 form of iron, back to +2. So if you lose the ability to convert your +3 iron and reduce it to +2, then that means you cannot you can't carry oxygen, right? Well, the red blood cells not only requires these enzymes to convert your iron back to the ferrous form, but these enzymes are also important for helping red blood cells become more resistant to oxidative damage. And because you lack this enzyme, you can't repair oxidative damage. Remember redox equations, reduction, oxidation. So this enzyme is responsible for giving up an electron, again helping make your ferrous form of iron, but also helping you to balance oxidative damage inside the red blood cell. So the red blood cell accumulates far more damage more rapidly, causes denaturation of proteins. The red blood cells can't get through the reticular fibers. They are more quickly removed from circulation. Now, anti-malarial drugs can cause profound hemolysis in people of African and Mediterranean descent, and here's the reason why. They cause anti-malarial drugs. The way they work is by causing a lot of oxidative damage. And if you take into consideration that a large part of the population has this G6PD deficiency, you're adding insult to injury. You're giving them drugs that will help them with the malaria, but then you're causing a lot of oxidative damage, not just to the plasmodium, but to the red blood cell, and then they can't repair that oxidative damage. So primaquine or quine is an example of an anti-malarial drug. Now, my father just passed away, and one of his favorite TV shows was MASH. And there was an episode of MASH where Klinger, remember he was the cross-dresser of the show, Klinger was given an anti-malarial drug, and he ended up having hemolysis. And the doctors were trying to figure out why. And it took them a while to realize that Klinger was of Mediterranean descent and had G6PD deficiency. So that was a whole episode. Now, getting back to Hannibal Lecter. These people with G6PD deficiency have to be careful with the types of food they eat, because some foods can cause more oxidative damage, which means then their red blood cells would become more damaged and more likely to be removed from circulation. Fava beans is one type of food that has high oxidative properties, and that's why I had that little clip of Hannibal Lecter. Now, some students like to see the overall metabolic pathway that's affected, and I do not want you to memorize the pictures on this screen. But if you look carefully at them, what you are seeing is a shuttle of enzymes that help shuttle along an electron. And again, the electron has to come from an enzyme, and it donates it to either reduce your iron back to the +2 or help with oxidative damage. Now, G6PD deficiency was the problem. G6PD, the gene, is found on the X chromosome, and if you look at the top right picture in black and white, or flow chart I should say, you see that G6PD, the glucose 6-phosphate dehydrogenase enzyme, is responsible for converting glucose 6-phosphate into the 6-phosphoglucose-6-lactone. But when it does this, it takes an electron and gives it to an electron carrier. So NADP gains an electron in this process and is converted to NADPH. And then NADPH will give its electron to an enzyme called glutathione. And glutathione is important in oxidative and reductive processes. And you can see this glutathione reductase and glutathione peroxidase. Very important with fighting oxidative damage. So if you lack G6PD, then you cannot take off this electron. And all of your enzymes that help maintain the levels of oxidative reductive damage cannot do their job. You're basically stopping this whole process. So the free radicals build up, and this leads to oxidative damage, and then the red blood cell membrane lyses from all that damage. Again, G6PD deficiency. You lack the enzyme to process glucose, but it is also a step where an electron is removed and given to electron carriers, and ultimately enzymes that help fight oxidative damage, free radicals. The damage builds up, and then the red blood cell is removed from circulation. That concludes our overview of many types of anemia. I hope you enjoyed your 4 sessions on erythropoiesis.